right. It's my great pleasure to be with Dr. Ewan Birney today at the hallowed grounds of the Mendel Museum here in Brno, Czech Republic. Welcome to the NEXUS Podcast. How are you doing?
Very good.
Yeah. So you have been a person on my mind since graduate school. I mean, an icon in our field of bioinformatics, biomedical informatics, and now at a place where I believe is also an icon in disseminating resources that are important for everything from education to how we do research in informatics today. It would be great to start off a little bit about yourself, and I know this is probably a complicated journey, but how you got to EMBL and the European Bioinformatics Institute. And before that, what do you do there actually?
Yeah, so my current role, I'm Interim Executive Director of EMBL. EMBL is rather like CERN, so it's the European Molecular Biology Laboratory, and it's the way in the 1970s Europe did federal research. They set up a treaty organization, so we're like a little country. We're owned by 29 other countries with an associate member of Australia. We operate as if we're embassies in 6 different countries. And our goal is to do research, service, including data services, which you mentioned, training, technology transfer, engagement with industry, and coordination across Europe.
That's tremendous. And to my knowledge, it's been focused on essentially everything related to biomedical data science, of which the recent thrust is our interest here is in Exposomics and in the NEXUS program is NIH US sponsored. Mm-hmm. How do you feel that this feeds into the things that EBI and the associated laboratories are developing? And can you tell us a little bit about the vision there?
Sure. Well, I think there's 2 strands in this. So let's do the data strand first, right? Which is at EBI, we hold the most comprehensive set of data resources anywhere in the world. And you'll know the Human Genome Reference and the Human Genome Set, Gene Set, and all of that. That comes from us. People who use that either are using our resources directly, or someone else has taken our information, packaged it up, and you're using that sort of second phase of information. Now, there's more to life than DNA, as you know, and we've been long tracking all the components from DNA. So the protein, the RNA sequences, the protein sequences, the protein structures, but also the small molecules. And the small molecules here are metabolites, and you can think about those inside of the cell, but also, of course, outside of the cell. And outside of the organism itself is what we often call exposomics, though, of course, that's a bigger field than just the chemical exposome. So there's always really been a real sort of alignment of people who've been looking at small molecules inside and outside of organisms of all sorts, humans, plants, where we are not, we're very Catholic in our taste of organism. So we will study all of those and store them. We've long had a database called metabolites, which allow the storage of that. Although that sounds as if it's focused on internal metabolites, it's actually absolutely fine for storing also external samples. Now, the other strand is, over the last 5 years, EMBL as a whole, the whole organization, has been sort of challenged and successfully stepped up to that challenge of stepping out of the lab. So this is studying life not only in the lab, but also outside, these very big surveys. We had an incredibly successful collaboration with TARA, the French research ship, which went across all the oceans and sampled many places across the globe, doing imaging, metagenomics, of course, genomics, but also this chemical assessments, so chemical studies. And that has been followed up with TREC, transversing Europe's coastlines, where the TARA ship went alongside the coastline of Europe. And we have an EMBL mobile laboratory that went alongside the ship on the coast. And the new thing we've added there is high pressure freezing and microscopy in the truck. So the truck it's like one of those transformer trucks. It kind of comes out. It's amazing. And that's allowed us to capture organisms kind of in situ not only as DNA, which everybody thinks is boring. I don't think it's boring, but also as life kind of frozen just in that moment. And that's given us huge insights. Now, we're going to, we hope, do the rivers of Europe coming up, fingers crossed. The Great Rhine, the Danube, which is so close to here, and other rivers and lakes across Europe. And that will really help round this out. But there, even more strongly, you get this question of chemicals. Of where are the chemicals, just where are the chemicals in these rivers? What sorts of chemicals are they? I know that there's a big focus on human health, and that's totally appropriate. But in many ways, you've got to think of this as an ecosystem. What are the chemicals in the ecosystem? Where did they come from? What are they impacting? So that overall view. Now, there's lots of other cases where we're also looking into mechanistic biology inside of the lab often, both with Drosophila and with mice and with other things. So we do have a kind of quite a nice suite here. Data, large-scale surveys of European areas, and mechanistic experimental studies.
It's truly amazing, the resources that are coming on board. A model for us in how to do surveillance. I totally agree. I think one of the challenges for exposomics is that we have these new technologies to measure in humans, but attribution is going to be a challenge, and figuring out where these things are coming from to eventually, better human lives. It's something to think about from the data perspective, since a multitude of different projects with the common theme of collection of data and driving and enhancing experimentation. For us as in exposomics, one daunting challenge is how to define and how to initiate a project. Like, what are we going to measure, and eventually how do we catalog and deliver it? Do you have any insights on how this type of process is initiated at EBI or EMBL in general, about these sort of large, big mission projects and how to get started, how to make sure they're successful?
I mean, I think there's all sorts of different takes on this. There's a kind of almost social take, what are you trying to do? How do you get scientists together? And then there's some very technical takes and some very important takes. Let me maybe do them in this order. So I think it's obviously the case, because the scope of what you could do is so big. You immediately are confronted by huge opportunities, huge possibilities. Everything comes with a price tag. It's very tempting to try and do it all. You'll never. That's never financially possible. Even for many good reasons, you've got to decide what you're actually going to do. What's your North Star? What am I trying to achieve? And I mean, the two things which I've noticed about successful projects, obviously you have to put together the right consortium. It has to basically work logistically and technically. And that very, very practical business of, well, how much does it cost? How can we flow the samples? Who's going to do what? These things end up being a far more important thing sometimes than people realize. But of course, the overall design, you have to think about, well, in some sense, talking as an epidemiologist, you have to think about, what's my sampling frame? What am I actually going to look at? And that has to connect to how you think you're going to do the analysis. Now, you don't have to have it completely worked out, but there has to be a kind of a good enough sketch across the board. And then just to dive into this, never-ending problem, expense and resolution versus cost. It is slightly intellectually boring, but nevertheless, it's true that you just need a lot of samples for many of these things. So there's a theme here right now. I've been banging the drum for this. I mean, it's very dangerous to ask a statistician or someone who does analysis, how big should a sample size be? Infinite is the number you really want. But what we've noticed, and I've really seen this, of course, now kind of flipping hats to these big human cohort studies, where I think previously people thought that 10,000 was a big number. But, actually when you get to 100,000, you are in a very different place. And I think what's interesting there is, of course, some cohorts go up to 500,000.
Yeah.
And definitely I feel there's a point where you get traction on a lot of problems at 100,000.
Yeah.
Obviously 500,000 is better. Obviously there's some interactions where we estimate some things we'll only see at a sample size of 10 million.
Yeah.
But 100,000 does feel like, for many, many studies on humans, 100,000 humans seems like a good place to start.
Yeah. Yeah.
And then just to say the obvious, but again, in all of this analysis, it's incredibly important that you're consistent with the sampling. Again, in the TREC project, this is not about humans. This is about locations across the European coastline. An incredibly important thing was that you were using the same protocols at every site. And that in itself is transformative, because you can start to truly compare data 100%. Between this. So that kind of lockdown of the protocols, understanding that. And then coupled with that is you need good metadata tracking, good ontologies that represent the metadata, good ontologies, again, that represent the results of the analysis as well. So you have kind of metadata ontologies to describe your samples and describe your data. And then you'll also have a sort of knowledge ontologies to describe the outputs of your analysis. And in this modern world, those kind of data engineering components, data logistics, knowledge logistics is as important as the kind of sample logistics that go on.
Yeah.
But, it's definitely doable. Let's go do it in a number of locations, and, just understand the world we live in.
It's super interesting. I think there are many parallels we could draw with in exposomics of getting that standardized assay measurement, the same measurement across all these different cohorts, and eventually going with large samples, longitudinal, because the effect sizes are relatively small and when looking at one of these factors by themselves. So you're absolutely right. We need to start combining. But one place where I'm super excited that you are making a huge impact potentially for our field of exposomics is reconsidering how we look at time in these cohorts with the use of artificial intelligence. I would really love to talk to you about this amazing paper you guys recently published. You guys, your amazing team there that you've collaborated across Europe with on a creative use of AI using these fantastic inspiration from the large language models. Especially, I think, in helping us understand the trajectory of disease across different place and time. Can you give us a summary about this work?
Thank you so much for those kind words. And you're right that this has been a great piece of work for me. So, my day job is sort of strategy and management and money and all of that important stuff. Sort of important things. I kind of have the privilege of still being research active and having a research group. And so this is really about that research group. And this is work I've done with Maritzka Stung at DKFZ in Heidelberg and Soren Brunak in Copenhagen. So, we're really bringing AI to epidemiology. That's what we're doing here. And like you said, we were really inspired by the success, the remarkable success of this generative pre-trained transformer model. I always get slightly annoyed that we don't emphasize the attention network component in that kind of phraseology. And really, I feel that it's, the transformer component is obviously incredibly important, where there's a kind of effectively a state space or an embedding space at every point. But what is the real, I think, eye-opening ability of AI are these attention networks, so this data-driven model discovery, which is just remarkable. So inspired by that, we brought that kind of technology to epidemiology. And actually, at one level, it's very similar. So in language, you have a token of words or parts of words. And the goal of the transformer is to predict the next token. And as you know, this amazingly can feel, to a human, it feels like it's talking to you. This simple business of, very deep prediction in the sense of very context aware prediction. So at one level, healthcare is the same. We have a series of tokens over someone's life, and the tokens are ordered, and what would be really interesting is predicting the next event you'll have in hospital. So that would be when you next go into hospital, what token would you be given by the doctor? But the big issue there, or the big difference with language, is time is really important in healthcare. And here's two sequentially identical examples. You had a heart attack. The next day you were diagnosed with pneumonia. That's really bad for you. That is a very bad indicator that you're in a bad way. But maybe you had a heart attack. You recovered. 2 years later, the next healthcare event in your record was pneumonia. Sequentially, they are the same, but in practice, there is a huge difference. And so it was really important to give our model access to that time component, but also the ability to predict the time component. So we're not only predicting the next event, but we're also predicting when that next event is going to be happening as well. Now, due to the steps forward in AI, we trained that up as when you know what you're doing, it's easy, but it's the when you know what you're doing bit, which is the complicated bit. We trained in this wonderful UK Biobanks, so 500,000 Brits, includes both my parents. So,
no kidding.
So 500,000 Brits, living their wild British life. And tested them with holdout data sets, zero-shot data sets in the UK. But we always had a bit of a sneaky, have we effectively learned something specifically about the UK Biobank or about the UK healthcare system? So we took the model with no changes in parameters. So a zero-shot test in the AI speak. And we took it to our colleagues in Denmark. You can get responsible access to the all Danish healthcare records, or many Danish healthcare records in different types. We happen to have 1.9 million Danes, so that's a fair whack of Denmark. It's not all. With our colleague Soren Brunak, and there we could test it in Denmark in a zero-shot way, where the model had never seen any piece of Danish data. And it performed very well in the UK, as well as any epidemiological model that has been used in the clinic so far. But we were able to do all diseases at once, rather than most epi models will do one or two diseases at the same time. And when we took it to Denmark, it only lost a small amount of performance when we assessed it in Denmark. So that was really reassuring that we were discovering things about patterns of human biology. I mean, obviously Western, OECD style healthcare, modern healthcare may not translate in the same way, but at least between the UK and Denmark, you did get this generalization, and gave us a lot of reassurance that we hadn't, our model wasn't taking shortcuts in the UK data. So that was very exciting. We then had a wrangle with some reviewers, the usual scientific back and forth. But it got published a couple of weeks ago. In the meantime, we've been working with colleagues. Andrea Ganna, who's at this meeting, is looking at it in Finland, in Finnish data. And we're really excited about adding so many different things, including exposures of all different sorts.
Super exciting. I'm really excited about kicking the tires. I think and all your code and data is out there for us to play with.
I mean, all the code is the data.
The data's out right there.
The data is in UK Biobank, and actually, an interesting aspect of this is that the weights we've for as an abundance of caution, we've said that the weights are potentially identifiable. And so the weights, you have to go back to UK Biobank to get the weights.
I would have to say also kudos to the folks at UK Biobank for providing such an amazing resource for the world to use.
I mean, I think, they have been remarkable. And, two things about that. Firstly, the cohort has been very well designed and thought through, and how they've done the phenotyping and the baseline. And as you know, that what that's done is it's crowded in investment from around the world in all sorts of different ways, where different groups have come and actually brought their technology to the UK Biobank and done that. I'd also say, by the way, the Danes and the Finns are the two other places which really have this incredible tracking of inside of their healthcare system, really, over and above anyone else. And the Danes were started in 1977. So the Brits, the Danes, and the Finns are, for me, three of the places. There are other places, but they're three of the places that truly lead in this kind of, huge scale cohort.
As an American, I hope that we can catch up to that, and I think it's truly important for human health, especially when we start to integrate things like exposome and genome and proteome. As you know, as we heard, the UK Biobank and these other biobank scale cohorts are starting to get a lot of multi-omic information that tells us also a lot about disease. I'm sure you're thinking about ways of integrating those data, if not already. Can you give us some tips about what we're going to see later?
Yeah, so I think, again, the UK Biobank, because of the way it's constructed here, is the candy shop. And it's remarkable the kind of things you can do. We've got really excited about retinal imaging, OCTs. So this is eye imaging. They also have cardiac and brain MRI on 100%, on 100,000 people. In terms of proteomics, they have some Olink assays, blood-based Olink assays. Actually, the one I'm really interested in is the Nightingale NMR metabolomics assay, because they have that on 500,000 people. And that really gives you the ability to get in there. I know NMR, I know many people here at this meeting is very mass spec-y, but this very simple business of sample size is incredibly important. So I already there's been some papers about them, and it's been pretty remarkable. I'm really excited about integrating that into the AI. Now, genetics, I think, is interesting. You can add genetics in as a predictor. But as you know, there's kind of Mendelian randomization ideas, or these ideas of using genetics really as a holdout data set for you to understand causality and understand, the order of events. And, we're really leveraging the fact that the genome doesn't change over someone's lifetime. So when we see correlations with the genome, we know that it must be the genome out, not anything coming back and altering the genome.
So as you know, AI has been fantastic, especially for using these models for multiple tasks. This idea of a foundation model that could provide you different ways of using your AI model. Have you been thinking about this idea of foundation models and how to use your new UK Biobank, Danish and Finnish model here to build a foundation for new waves of epidemiological research?
I mean, at the moment, our models are still small enough that we can just retrain from scratch for most tasks. And actually, I prefer sort of intellectually retraining from scratch. Of course, what you do is you learn what architectures work and why they work and this sort of thing. But I think what is true is that this kind of compositing models, so for example, these imaging models, the OCT images and those things, it seems very hard to imagine to train a good model end-to-end of both the trajectories and the OCT representation at the same time. And so I can imagine compositing different models together. So it's a slightly different zone from foundational models a la LLMs, where you think about this like incredibly big parameter space, which you then put on different outputs. But I think what we need is a lot of innovation and a lot of methods development in this. That's one take on this.
I'm so excited to see what the role the exposomics could play in this new future. And with that, we will conclude. Please follow us on social media, YouTube, X, LinkedIn, and our website, nexus-exposomics.org. Thank you so much, Ewan, for joining us today on Nexus.
My pleasure. Thank you so much indeed.